examining the mechanism of FAR-R in promoting increased resistance of Staphylococcus aureus to antimicrobial-free fatty acids on human skin. Staphylococcus aureus is a gram-positive bacterium that forms clusters of cocci. Approximately 30% of healthy adults are asymptomatically colonized, preferentially in the nares, but also on the skin. Typically, the colonizing strain most commonly causes subsequent infection. Consequently, S. aureus is opportunistic and utilizes immunocompromise to cause invasive infections such as soft tissue abscesses, lower respiratory infection, pneumonia, endocarditis, bacteremia, septicemia, osteomyelitis, and cystic fibrosis complications. Burden on human health was also increased with the introduction of antibiotic resistance in 1961 through the acquisition of the mobile element SCC-MEC. Methicillin-resistant S. aureus, or MRSA, is prevalent in two forms. The most common community-acquired, or CA-MRSA, is USA 300, possessing the arginine catabolic mobile element ACME and SPG for polyimmune resistance. On the other hand, the most common hospital-associated, or HA-MRSA, is USA 100, where some possess intermediate susceptibility to vancomycin and lack ACME. USA 300 represents the strain we will be using in vitro, while USA 100 represents the strain of our clinical isolates. As previously mentioned, skin/soft tissue infections are important and represent 90% of all CA-MRSA, indicating a need to examine S. aureus colonization of the skin. The skin represents the initial defense against pathogens, utilizing keratinocytes with tight junctions, antimicrobial peptides, immune cells, and a competitive microbiome to protect against infection. However, it is the acidic pH of 4.5 to 5.9 that is of particular interest. As observed in the diagram, many mechanisms contribute to pH, such as the conversion of filigrin to uronic acid, the NHE1 sodium proton antiporter, proteases, and enzymes. Today I'm going to be focusing on the conversion of phospholipids and ceramides from sebum into unsaturated free fatty acids, or UFFA, by commensal secretory lipase A2, or ceramidase. These UFFA are toxic to bacteria through membrane insertion that causes subsequent instability and increased fluidity. This protein leakage, electron transport chain interference, and dysregulated nutrient uptake cause cell death. However, these UFFA aren't always toxic. S. aureus has used these lipids to its benefit in its first defense against toxic UFFA. UFFA, such as sapienic acid in sebum and linoleic acid on the mucous membrane of the nares, cross the bacterial cell wall/membrane and enter the fatty acid kinase, or FAK, pathway. Instead of interfering with the membrane to cause increased fluidity and detrimental cell leakage, UFFA are bound by FAK-B2, then phosphorylated by FAK-A before they enter either the FAS2 or PLS pathways for conversion into and incorporation as bacterial phospholipids. However, what happens when the maximum capacity of FAK-A to incorporate these UFFA is reached? This question led to the discovery of a novel UFFA detoxification mechanism in S. aureus. Previously in our lab, a novel TET-R family regulator called FAR-R and a resistance nodulation division R&D superfamily efflux pump called FAR-E were discovered. USA 300 was exposed to high levels of linoleic acid, which selected for a variant with increased resistance. This variant, called FAR-7, possesses an H121Y substitution in the ligand binding domain of FAR-R, with increased UFFA resistance and constitutive FAR-R expression. FAR-R can be shown here bound to the PAL1 and -10 operator sites for autorepression. Based on existing models, we suspect that FAK-A metabolic products interact with the ligand binding domain of the TET-R family regulator FAR-R. FAR-R then binds as a dimer to the divergently transcribed intergenic segment with FAR-E at 3 operator sites possessing the TAGW-TTA consensus sequence. We suspect the binding of the helix-turn-helix motif of the N-terminal DNA binding domain of FAR-R at the palindrome PAL2 operator site induces FAR-E expression, unlike a typical TET-R family regulator. However, the specific metabolite that causes FAR-R activation remains unknown. Although resistance nodulation division fatty acid efflux pumps have been widely characterized in gram-negative species, they remain uncharacterized in gram-positive species. Additionally, the ligands, pathways, and mutations responsible for FAR-R function remain unknown. Thus, we have two hypotheses. First, we hypothesize that once maximum UFFA incorporation capacity is reached, a metabolite from the fatty acid kinase pathway forms a complex with FAR-R for activation. Second, we hypothesize that S. aureus, like the ACR system in E. coli, is subject to selective pressures in vivo that result in FAR-R mutation and subsequent de-repression of FAR-E. A previous electrophoretic mobility shift assay from our lab was performed and demonstrated FAR-R was unable to bind a minimal 24-nucleotide PAL2 probe, shown as OP4.1. A lack of binding was demonstrated by the absence of complex formation for the OP4.1 segment. The first two sites represent FAR-R binding to its autorepressive sites, while the third represents the activating site at PAL2. This suggests FAR-R nonspecifically binds a longer probe, while we suspect a small molecule ligand is required to be present in the EMSA as a cofactor to specifically bind the minimal probe. Two possibilities exist based on these data. One, an acyl phosphate, a metabolic product from the FAK pathway, binds directly to FAR-R. Or the acyl phosphate binds as a complex with additional proteins to FAR-R. This is similar to another activating TET-R family regulator called DHAS from Lactococcus lactis that requires the DHAQ protein ligand to activate the dihydroxyacetone operon. Biology search in the NCBI database returned a 26% identity to the structure of the dihydroxyacetone kinase regulator DHSK from Listeria monocytogenes. The PDB file of DHSK was then used as a template to predict the structure of FAR-R using Modeler 9.25. A CyPred secondary structure prediction was used to increase the accuracy of the alignment. 20 models were created, and the model with the most negative dope score was selected. The process was then repeated with the dimer structure. Since DHSK requires a small molecule along with a protein for activation, the structural similarities support the hypothesis that a cofactor is involved. This means that FAR-R could potentially bind to a complex involving any of FAC-A, FAC-B2, ATP, acyl phosphates, and linoleic acid. More work still needs to be completed with the modeling. The BIPSI server will be used to assess the probability of FAC-A and FAC-B2 binding, while the PISA server will scan for biologically relevant interfaces. My next steps are to repeat the EMSA with the minimal 24-nucleotide PAL2 probe in the presence of FAC-A, FAC-B2, ATP, and linoleic acid. If a shift is formed at Site 3, then it can be confirmed that a metabolic product or complex from the FAC pathway is involved in the activation of FAR-R. From here, we will perform size exclusion chromatography, or density gradient centrifugation, followed by mass spectrometry to identify which proteins are forming an activation complex with FAR-R. As mentioned earlier, in vitro selection of USA 300 with linoleic acid resulted in discovery of FAR-7, a variant of FAR-R with an H121Y substitution, demonstrating increased UFFA resistance and constitutive FAR-R expression. Bioinformatic analyses have revealed several prominent globally distributed FAR-R amino acid substitutions in a USA 100 background. 11 isolates have been collected that possess a C116Y substitution in the C-terminal ligand binding domain of FAR-R. The isolates are from the Broad Institute at Harvard and are representative of a wide range of locations such as sputum, bloodstream, and nares. Since the position of the mutation and substitution for a tyrosine residue are similar between the H121Y and C116Y mutants, we suspect they will share a similar phenotype of increased UFFA resistance. Total MIC of the FAR-R C116Y isolates in the presence of linoleic acid demonstrated increased growth shown by OD600 and UFFA resistance compared to USA 300 for all isolates except M0049. Sequencing revealed that M0049 does not possess the C116Y substitution, thus it possesses wild-type FAR-R and should display UFFA resistance comparable to USA 300. 4 isolates with an E160G substitution in the ligand-binding domain of FAR-R come from cystic fibrosis CF patients co-infected with Pseudomonas aeruginosa. However, 60 genomes were found with this mutation from Israel, the US, and UK. CF patients co-infected with MRSA and Pseudomonas demonstrate poor lung function and worse outcomes. However, Pseudomonas produces a hydrophobic small molecule 2N-heptyl-4-hydroxyquinoline N-oxide, or HQNO, that incorporates into the MRSA membrane to increase fluidity and allow antibiotic penetration into the biofilm. We suspect that since the molecular structure and mechanism of toxicity of HQNO are similar to UFFA, the FAR-R E160G isolates evolved increased resistance by utilizing FAR-E to efflux HQNO. Additionally, 3 isolates with a P165L substitution in ligand binding domain of FAR-R have been collected from Mississippi and include MRSA from a CF patient in Georgia, a CF patient in Cincinnati, MRSA from Denmark/Egypt, and cheese-associated MRSA from Austria. However, 33 genomes were found with this mutation from the US, Denmark, Egypt, and Scotland. These isolates are of particular interest because the T688 spot type has been implicated in potential chlorhexidine resistance in bovine mastitis in Egypt and hospitals in Kuwait. Chlorhexidine is a disinfectant used in both healthcare and animal agriculture. Since both mutations are located in close proximity to each other, we expect they will share similar resistance phenotypes. An initial MIC of the FAR-R E160G and P165L isolates in the presence of linoleic acid demonstrated variable growth in UFFA resistance. Both USA 300 and USA 100 were used as negative controls. Only HP16, HP67, and HP99 from the E160G isolates, along with DAR 948 from the P165L isolates, showed increased growth in UFFA resistance compared to USA 300 and USA 100. Sequencing will be performed to confirm the presence of the desired mutations. DAR 1874, indicated above, was also included as it possesses an H43Y substitution in the DNA binding domain of FAR-R, because we were curious to see if altering the DNA binding ability of FAR-R altered UFFA resistance. 4 strains with a GAG duplication at glutamic acid 93 and FAR-R are from the nares of patients from the Broad Institute at Harvard, but have also been found in vancomycin intermediate S. aureus, or VISA, from Japan, MRSA from human skin in Japan, and food-related MRSA from China. However, 21 genomes were found with this mutation from Japan, China, the US, and the UK. Initial MIC of the FAR-RGAG duplication isolates in the presence of linoleic acid demonstrated increased growth in UFFA resistance compared to USA 300 and USA 100 for all isolates. The growth was also comparable to FAR7. Sequencing will again be performed to confirm the presence of the GAG duplication at position 93 in FAR-R. In summary, the FAR-R C116Y and GAG duplication isolates showed increased UFFA resistance, while the results for E160G and P165L were variable. A Western blot using E. coli DH5 alpha lysates with PLI FAR-R variants was performed to determine if the mutations caused de-repression of FAR-R and constitutive expression like FAR7. Empty PLI 50 was used as a negative control. Only the C116Y substitution showed an effect on FAR-R production, while the others did not. After confirming the sequence of the FAR-R variants, my next steps including one of each of the PLI50 expression vectors in E. coli into a lab strain of USA 300 with a transposon knockout of FAR-R. Then an MIC will be performed in the presence of linoleic acid to determine if each amino acid substitution alone is sufficient to cause increased growth in UFFA resistance. Both the original isolates and USA 300 PLI FAR-R constructs will be subject to MICs with HQNO, chlorhexidine, and palmitoleic acid to determine if FAR-R provides resistance to alternative antimicrobial compounds and UFFA. Finally, the FAR-R variants and the FAR-E promoter will be cloned into PGY-Lux, transformed into the FAR-R transposon knockout of USA 300, and used in a luciferase assay to determine if FAR-E expression is altered by changes in FAR-R. MRSA still represents a significant burden on global health, representing 74% of global S. aureus cases and possessing a bacteremia mortality rate of 30 to 50%. Understanding the mechanism by which FAR-R is activated and induces a flux of toxic UFFA will provide insight into potential targets to prevent colonization. With persistent antibiotic resistance, it may be beneficial to shift treatment from curative to preventative. Additionally, mutations have arisen in a clinical setting that allow S. aureus to evade innate immune defenses such as UFFA, demonstrating an important example of in vivo selection and adaptation to both hosts and potentially to competing microbes such as Pseudomonas. Potential resistance to compounds such as HQNO will provide insight into alleviating the burden of disease in patients with cystic fibrosis, while examining chlorhexidine resistance will benefit our understanding of sanitization procedures. Thank you for listening. These are my acknowledgments.